want to see what you are doing. Yeah. Can you see what I'm seeing? I'm displaying on my screen. Can you see that? Please, can you read what you have here? What's the full meaning of DIY? Do it yourself. So, once again, good afternoon, everybody. I want to really believe that you must have learned introductory to bioinformatics in your first year. And if you have done that, this is just going to be a continuation of what you have learned so far. Time is going to fail us if I start telling you about or start asking you what bioinformatics is all about. But if you want to know, you should know that I said bioinformatics is not just a course, it is a career, it is a profession, it is a discipline. And what is that discipline coming to do? It is a discipline that combines computer science, statistics, mathematics, biological sciences, biochemistry, and some other parts of other disciplines. They are coming to know more. They want to know more about biological data. And biological data that we'll be dealing with is these are informational molecules like the nucleic acid, DNA, RNA, and so on and so forth. And this is going to be in furtherance to that, and that is why how applicable is bioinformatics to you, to your career as a microbiologist. And that is the reason why that today this is being presented to you. In this presentation, it is going to be an expanded step-by-step laboratory manual. It's not going to be only that, it is going to be with visuals, it's going to be with images, it's going to be with case studies and examples. And why I call it DIY is that while you are watching it or you are seeing it displayed on the screen, you are expected to be following it as it goes. Don't forget that this is just the first part of the presentation. The second one is coming, that you'll be left alone and you are going to work in pairs. So follow me as we go together. Now, the first thing is step one, which has to do with how can you retrieve a bacterial genome from NCBI. The tool you are going to use is already displayed there. You are going to make use of NCBI genome databases, and these databases is where you have free sequence DNA sequences. Not only DNA, you have other sequences as well, and that is what you have. And what you are going to do is to search for Escherichia coli complete genome, and by the time you will be doing your classwork, you will not be searching for Escherichia coli again. You will be searching for a specific microorganism in which is going to be chose by you. Then you are going to apply filter by ref sequences and complete genome entries, and the downloadable form of all these genomic data sequences is going to be in faster format. And you can also make use of the gene bank accession number. So after doing that, this file will contain your DNA sequence and annotation. So whatever you must have done, immediately when you get to the last step, you will screenshot this. You screenshot your results, that is the nucleotide basis you must have gotten. You screenshot it and you go and paste it on ChatGPT, and you will prompt GPT to interpret that result for you. It is going to also help you to do that. And just like I said at the beginning of this presentation, that this presentation is harmed with is provided with other features, and when you check the right side of this slide, you are going to see some kind of images there that can actually guide you of what to do at some particular time. So if you see, you will see that you have the NCBI genome browser, and inside it you have Escherichia coli complete genome. That's what you are going to type there, and you just hit your search button, and what the next thing you will see is that the Escherichia coli complete genome is going to be soon going to be displayed, and you have it in two formats, in form of .fna or .gbk. So whichever one you want, if you click on .gbk, that means you want it to be in gene bank, and if you click on .fna, that means you are retrieving your file in form of FASTA. Step 2. The step 2 today, you are going to use step 2 to predict antimicrobial resistance by the use of a card-rgi. And don't forget what antimicrobial resistance is all about. This is the ability of microorganisms to undergo a kind of mutational changes or molecular changes, whereby you see some set of antibiotics that they are not going to be effective against all this set of microorganisms. And they always do this by using a lot of molecular mechanism by undergoing that. That means the antibiotic that you have been using that has been so effective against them is no longer going to be effective. And as a microbiologist, if you want to discover whether the organism you have is going to develop antibiotic resistance in the nearest future, well, what you need to do is just to go and predict antimicrobial resistance by the use of a card. And the full meaning of card is a comprehensive antibiotic resistance database. So, and just like what you have the tool. The tool is called card-rgi. And what does it do? It identifies known antibiotic resistant gene. So what you will do when you get to that database is to upload your genome file. It could be in form of .fna or .gbk. So after that, you select whichever one you are looking at. When you get to that site, the site is going to group the antibiotic resistance, is going to group it into 3 for you. It is either perfect, treat, or lose. So whichever one you hit is going to be a reliable result for you. Then you click on submit job button to be able to identify the types of drug that that microorganism is going to be resistant against in the nearest future to come. And it is also very possible that you can also predict continent by continent, region by region. So you can equally use card to be able to discover whether they are going to be resistant or not. And thereafter, after submission of your job, the next thing is to review the output. And how will you review the output? By their gene name, by their drug class, and by their match type. We are going to document this, and which is very, very important for you. Then on the right hand of we have the image of what you are going to see when you open card-rgi. So what you will do is that the genome file, the file that you must have saved, you are going to upload it. Then you click on analyze. By the time you click on analyze, the result is going to be displayed. There's going to be ARO identity numbers, and predicted resistant genes, and the class drug is going to be there. Don't forget that the review, the output, just like I said, is going to be the gene. So these are the gene names. Then the drug class is also here. Then the match type is also here. That is That's about step 2. Step 3 now is to use 16S ribosomal RNA sequence identification by the use of BLAST. What does BLAST? What does it do? BLAST is the full meaning is a basic local alignment sequence tools, and that is the tool that you normally use to be able to discover the proximity in terms of relatedness, how similar microorganisms are and how dissimilar microorganisms could be. So you will make use of a BLAST to be able to do that. Then don't forget what we said in microbial evolution and ecology. You must have come across what is called WOESE, 3 domains of life, whereby the classification of organism was based on 16S ribosomal RNA. And I told us then that RNA was more reliable than DNA because at certain points in time they discovered that over some long period of time that ribosomal RNA is still more stable than even DNA, and that's why one of the most reliable way of identifying any microorganism is by the use of their 16S ribosomal RNA. It's just like when you have the whole organism when you have the whole organism, that organism you now cut part of the fingers. The type of fingers could actually help you to know the group of that organism. For example, the hind leg of a cow is quite different from that of a horse. But by the time you see it, you'll be able to know whether it is that belongs to the cow or it belongs to us. And that is exactly the same way that 16S ribosomal RNA, that is what it does. So BLAST is by comparing, bringing your sequences, that is your 16S ribosomal RNA sequences, you are comparing it with every other sequences that you have in the gene bank. So, and the tool that you are going to make use of is called NCBI BLAST, which finds similar sequences. So, the more similar they are, the more related they are. So, what you will do is on NCBI, when you click BLAST, on that NCBI, you click BLAST, then you are going to obtain the gene of 16S ribosomal RNA from the genome. So the next thing you do is to paste it into NCBI nucleotide BLAST search bar. And the next one is to be able to use that tool to be able to do microbial identification, species identification. And the top match, that is, if those organisms, your sequences, the more, if your sequences now has greater than 99% identity, that means that organism is very, very close to be. And when you look at this other side, you could see what this person, what he has been able to do. What he did was to paste 16S ribosomal RNA in that NCBI BLAST, and he used that query. He used that query to be able to discover different types of bacterial species, which range from Escherichia coli, Salmonella enterica, Shigella commune, and so on and so forth. And by the time you now compare their similarities, the higher the number of their identity in terms of their e-values. What you look at is total score. Then you look at the query score, as well as the e-values, and you check the identity. And whatever results you must have gotten, you must take them down, and you go and make use of ChatGPT, and ChatGPT will also help you to be able to do that. I'm actually asking you to always make ChatGPT to help you to interpret, because by the time you will be doing this, maybe it's going to be during the practical exam, or it's going to be anywhere, you might not be asked, I mean, you might not be allowed to actually use ChatGPT to do that. But at least for a beginner just like you, it is better. And don't forget that this is a practical class where you are even expected to demonstrate some kind of perfection when it comes to, I mean, professionalism when it comes to your interpretation of this. So, and from there, you can even ask ChatGPT to do a lot of other things for you in terms of their similarities, in terms of their relatedness, in terms of any other thing that you might be asked to do. Then the step 4 is how can we build a phylogenetic tree using what is called cluster omega or ITOR. By now, ITOR should not be something that is going to be new to you, because you must have used that effectively in your first year. So what you will do for whenever you come across what is called phylogenetic tree, what that should tell you, phyllo means evolution, phyllo means from phylum, phylum means from origin. That is, if you want to talk about how closely their ancestor could be in terms of evolution, the next thing is to employ what is called phylogenetic tree to be able to know their level of relatedness. And the closer they are in terms of their similarity, the more closely they are together. So, but once they are far apart, so as you can see from this left, this right picture or image here, you will see that in terms of relatedness, Shigella is closely related to Escherichia coli. So you also see Salmonella. And the other one, in the other colors, those ones that are far apart, those are the ones that if you look at the branch of E. coli, Escherichia coli, and Shigella, you will see that they are too close, I mean, they are too farther apart. So once you get to this level, you could actually screenshot this and go and upload it as ChatGPT, and also ask ChatGPT to explain for you or interpret for you. So you could begin to ask the AI different types of questions, and it's going to give you. So what do? You collect multiple 16S ribosomal RNA sequences. That is, which is going to be your own sample, the one that you have collected, then plus other references. Now, something should come to your mind. Anytime you want to plot phylogenetic tree can only be plotted if you have more than 3, 4, 5 sequences. So if you have 2, for example, now if the sample you have a sample 16S ribosomal RNA from Shaki here, and you have another sequence from another site. So if those are your 2 samples, you know you don't have more than 2. What you will now do is to go to the database, go and look for references. So you go and get 3 more to add it to your own 2, and that is when you can now employ phylogenetic tree to be able to know how closely related they are. So, and you should be able to know that you collect multiple 16S ribosomal RNA sequences, which is going to be your sample plus other references. So the next thing is to go to cluster omega, and what you will do is to align the sequences. Alignment of sequences will tell you how closely related they are. So you align the sequences, and by the time you align it, after that, the next thing you do is to export it. You export it in form of a NECWIC format, and by the time you export it now, it is now going to be the next thing is to upload the file into ITOL. And the meaning of ITOL, who can tell me the meaning of ITOL? That is what? ITOL means what? Interactive tree of life. Okay, to visualize the tree, that is what you will use to be able to visualize it. That one will help you to be able to see it very well. Then you'll be able to see different types of relationship, and you'll be able to cluster. When you look at the image that you have here, you will see that it is appearing in form of a kind of a clustering of your isolates. Those ones that are very similar, they will belong to very close cluster, and those ones that are apart, they will stay too long from there. Then what you will do is, now you interpret your evolutionary distance among the strains. So what you will now do is you are going to screenshot the ITOL image, screenshot it, then you now list those organisms. You now tell the ChatGPT to interpret that result for you in terms of their evolutionary distance. So you are going to get a result, and that result is what you actually need. The next one is step 5. That is you want to analyze microbiome. Analyzing microbiome by the use of a special tool that is being used by microbiologists just like you. So we said it's going to be MG-RAST, and this is optional. That is, you can also use other tools to be able to do the same thing. So just like sometimes if you are looking for knife, and if you see tester, or you see plier, you could also make use of that tool to do whatever you want to do. So you will use this. So the question now is, what do we mean by microbiome? Microbiome is a community of different microorganisms that are living, or they are freely interacting together. Just like when you have the community of people, where you have different houses, people from different houses, they have their own level of organization where they interact together. They interact together in terms of their security. They come together anytime they want to take any common stand. So the same thing is also applicable or obtainable in microbiology. And what do you do? For example, now if you just collect soil sample and you want to know what are the types of microorganisms that you can see in the whole of that soil. Don't forget that soil is a diverse. Soil contains diverse microorganisms or organisms that are interacting together. So we make use of MG-RAST to be able to do that. The tool you use is MG-RAST, which processes and annotates, not only it processes and annotates it for you. Annotation means that it is going to tell you the function of each. For example, if the RAS is telling you that this is okay, in fact, when you look at the image on the right, you will see that you have the taxonomic analysis and the functional analysis. So RAS